Let's do a quick refresher on pH, pKa, and we'll also talk a bit about biological buffers. You've done this in general chemistry, but I want to bring that back because it's going to be so important to biochemistry and biochemical regulation inside the cell. So a quick refresher, what is pH? pH is a measure of the concentration of H plus ions. So remember that these brackets here, that is shorthand for concentration. So if we take the negative log of the concentration of H plus ions, we can calculate the pH. I also want to point out mathematically this is equivalent to the log of 1 over the concentration of H plus ions. These are the same. Negative log is the same as the log of 1 over. So that's the definition for pH. Again, some basic acid-base chemistry. Acids will ionize to form a proton or a hydrogen and a base. So the acid, and this is reversible, so the chemical equation can go left or right, right or left. So that acid can ionize to form that proton, that H plus ion, and a base. So looking at a real chemical reaction, we have acetic acid here on the left, which can ionize to form that proton plus acetate. And you'll notice these are both ions, so that's why it's an ionization reaction. So the chemical formed upon ionization of an acid is called its conjugate base. So this is the conjugate base in this chemical equation. Conversely, the acid formed when a base finds a proton is called its conjugate acid. So if we're starting with acetate, this would be the conjugate acid, acetic acid. Just a little bit of vocab there. I want to talk about the way this applies to water. So water at pH 7 will contain some amount of ionized OH and H3O+. This makes sense, right? Because to have a pH, you have to have some concentration of H plus ions, and we know the pH of water is hypothetically 7, so it tells us the concentration of H plus ions is 10 to the minus 7th molar. Okay, so when we talk about acid-base chemistry, we're often using this notation of H+. I want to point out that in an aqueous solution, so when there's water around, H+ does not exist. So let me say that one more time. Free H+ does not exist in aqueous solutions. Here's what really happens. When you have this hypothetical situation of two water molecules, one of them acting as the acid, the other one acting as the base, what it really forms is H3O+. So that free proton does not remain in solution as a free proton. Instead, it will bond to a water molecule to make this H3O+, which is called a hydronium ion. And then we do have a hydroxyl that does exist in an aqueous solution. So just know that we're going to write H+, but in reality it's H3O+, and water also gets involved in this acid-base chemistry reaction. And the name for H3O+ is a hydronium ion. Okay, so we're going to use this constant called the acid ionization constant, or abbreviated Ka, to describe the strength of the acid. So for a weak acid, the acid ionization constant is the ratio of the concentration of products to reactants for a chemical reaction at equilibrium. So let's say we have this hypothetical acid-base reaction where HA is going to be our acid, and then it dissociates or ionizes into our proton plus our base. So these are the reactants, or I'm sorry, these are the products, excuse me. So we have the concentration of our products divided by the concentration of our reactants, and then at equilibrium, we're going to have certain concentrations for each of these. We take that ratio, and that's how we get our Ka. So the key thing here is that the larger the Ka, the stronger the acid, because a strong acid is more likely to dissociate to make this proton plus base. So if we have higher concentrations of proton plus base, it's going to get us a larger Ka, and that tells us we have a stronger acid. All right, so now we've got Ka. We know what pH is. We are going to derive a relationship between those two. And this is going to be super useful, I promise, so just hang in there. So we just talked about Ka. We just derived that and said this is the definition for Ka. We're going to start by rearranging this equation. So just doing some simple algebra, we can rearrange it to get 1 over H+ on one side, 1 over Ka, concentration of base over concentration of acid over there. Now we're going to take the log of both sides. Why? Just trust me, because we're going to do it. So mathematically, this looks like this. So we have log of 1 over H+ is equal to log of 1 over Ka plus the log of A minus over HA. Okay, so remember that small p, like in pH, means the negative log. So thinking about that, what substitutions can we make? Well, remember the definition for pH, right? pH means the negative log of H+. That's what the little p means. And we also said that was mathematically equivalent to the log of 1 over the concentration of H+. And that log of 1 over concentration of H+ is exactly what we have over here. So we can take this term right here and substitute it out and put in pH. Now we can also define a new term that we're going to call pKa. So we're going to use that same lowercase p relationship to make the pKa. And that gets us to this equation right here, where the pH is equal to the pKa plus the log of the concentration of base divided by the concentration of acid. This is called the Henderson-Hasselbalch equation. And it's worth knowing, when you hear Henderson-Hasselbalch equation, you need to be thinking pH, pKa, and this relationship. I said this was going to be useful, so what do we do with it? We can use this relationship to predict a molecule's charge at a given pH value, which sounds super useful in predicting how biomolecules, such as amino acids, are going to be charged or uncharged at a given pH, which tells us what they're going to want to interact with. So how do we do that? So looking at this equation, here's one first truth about it. When the concentration of base is equal to the concentration of acid, we're going to have the log of those two, and if these two are equal, then it's going to be 1, right? One number divided by the same number gets you 1. When you take the log of 1, that equals 0. Plug that into your calculators right now. Prove it to yourself that the log of 1 is 0. So if this whole term is 0, then our pH equals our pKa. And what that tells us is we have an equal amount of both the protonated and the ionized form, an equal amount of acid and base. Okay, so for any acid, if the pH is greater than the pKa, then that ionized base form is going to predominate. And conversely, if the pH is less than the pKa, then that acid form predominates. Think about that for a second. Prove it to yourself. You can plug some numbers in, do some quick calculations, and you will find that this is correct. So how can we use that? Let's take acetic acid, for example, which is known to have a Ka of this, so that tells us about the ratio of acid to base at equilibrium. We can, from there, calculate the pKa, and we find that pKa is 4.76. So when the pH is less than the pKa, less than 4.76, we are going to have the acid form, the protonated form. When the pH is greater than the pKa, so moving past that 4.76, we are going to have the base form, that ionized base form plus the free proton. So again, the power in this, in the Henderson-Hasselbalch equation, is that we can predict which form this molecule will be in, the acid form or the charged ionized base form, if we know the pKa and we know what pH we're at. Some compounds might have 2 pH ionization events. So for example, if we look at carbonic acid, it has the first pKa of 3.77 that describes this first proton popping off. And then it has a second pKa of 10.2 that describes the second proton popping off. So again, knowing that carbonic acid has these 2 pKa's, we can predict whether we're going to get H2CO3, HCO3 minus, or CO32 minus based on what the pH is, knowing that pKa. So see if you can apply that for yourself. Here is alanine, the amino acid alanine. And we notice there's these 2 groups that are going to go through acid-base chemistry, becoming protonated and deprotonated. So I've given you the pKa's for each of these groups here. So one for the amine group has a pKa of 9.7. And then this carboxyl here has a pKa of 2.3. So given those 2 pKa's, predict the charge for each group, the amine and the carboxyl, at a given pH. So pH 1, what's the charge of the amine? What's the charge of the carboxyl? What's the overall charge of the entire alanine molecule? To do that for pH 1, 7.4, and 11, just make sure you're following along. We're going to move through and finish up with buffers. So a buffer is an acid-base conjugate pair that resists changes in the pH of a solution. So what does that mean? So for example, we have again our acetic acid and our acetate, and they will resist changes in pH. So there's this buffering region where you have a mixture of both molecules present in solution. And when you have this mixture of both molecules present in solution, you're going to get this buffering effect. So for example, acetic acid has this buffering region from a pH of 5.76 to 3.76. So in that range, it's going to be resistant to addition of acids or bases. You can add acids and bases and the pH is not going to change very much. So what you can see if we start at a very low pH, an acidic pH, where of course we would have this acetic acid form, and you start adding base, say some strong base, sodium hydroxide, you add a certain amount of it, and of course the pH goes up. And then you get into this buffering region where you're adding base, but the pH is not changing very much. And that's because you're in the buffering region where you have similar amounts of acetic acid and acetate, the protonated and deprotonated acid and base forms. And then once you get past that buffering region, when you add more base, the pH goes up quickly again. So in other words, acetic acid acts as a buffer. It buffers the pH. And a buffer is most effective at pHs near its pKa. So here's another plot to illustrate this. So again, we have our solution of sodium acetate. This time we're going to start at high pH and we're going to add acid. So if we had just pure water, no sodium acetate in it, we add that acid and the pH quickly drops because there's no buffering agent. If we have our sodium acetate, then we're going to get this gradual pH change in the buffering region of sodium acetate, again, because we have equivalent concentrations of sodium acetate and acetic acid, the acid-base conjugate pair. Here are the buffering regions for several common biomolecules. We've got ammonia in here, we've got a phosphate, and we've got acetate that we just talked about. So you can see how the pKa for each of these molecules illustrates its buffering region. So if you want to maintain, let's say pH 7, for example, the best buffer that you would want to use for that situation would be your phosphate buffer, because it has a pKa of pretty close to 7. So it's going to do a good job buffering in that pH 7 region. Whereas if you try to use something like an acetate buffer, it's not going to do a very good job at pH 7, because at that point you've already converted all of your acetate to its base form, and it's not going to buffer anymore. This is a critical concept in biochemistry because the pH of your body is so closely regulated. Because if the pH changes, you're going to have different groups becoming ionized or protonated in ways that you don't want, because that would change their interactions. So here's an example. The pH of blood is buffered by the conjugate acid-base pair of carbonic acid here and bicarbonate, HCO3-. The carbon dioxide produced in aerobic respiration reacts with water to produce the weak acid carbonic acid. So when you breathe, you're producing carbon dioxide. That is going to have some small interactions with water to produce carbonic acid here. So you're already producing carbonic acid because you have CO2 around. Carbonic acid then ionizes to produce a proton and bicarbonate. And carbonic acid has a pKa, two pKa values of 6.3 and 10.3. So that pKa, the first one of 6.3, is going to do a good job buffering your blood pH, keeping it close to physiological conditions. And finally, we're going to talk about some different pH areas in the cell and in the body. So we all know our stomach acid has a very low pH of 1 to 2, which is important because we need that acidic condition to digest our food. But this has a clinical application because when that stomach acid gets kicked back into our esophagus, that causes gastric esophageal reflux disease, or GERD, which is a huge issue for a lot of folks. So having the right pH in the right place is super important to prevent disease and tissue damage. Our blood pH is 7.4, which is what we're generally going to consider to be physiological pH, and that needs to stay at that value. Another application is inside of lysosomes. So we briefly talked about this, but just to reiterate, the cell can intake nutrients from outside of itself, and then it encapsulates them in a vesicle or a membrane-bound sac inside of itself. That sac then fuses with a lysosome that has enzymes that break things down and also has an acidic pH. So the pH inside of a lysosome is 4.8, a little bit acidic, which is needed to digest nutrients inside of the cell. So again, the key thing is that we need to understand pH and pKa because those will predict the structure of our biomolecules in different environments. We need to be able to look at an amino acid and know is it going to be charged or uncharged, because that completely determines how it will interact with the other molecules that are around it.